Well. Hello, everybody that's watching this. We are going to start the day looking at what we have in store for the week ahead. Hey, Evelyn. So I'm just going through what we have in store for the week. So we're going to look at bone physiology. Exam number 4 will be due at the end of the week, and that will cover chapter 16 and chapter 17. It will cover the neural integration of the autonomic nervous system, as well as the special senses, the eye, ear, and the gustatory and olfactory system. So that's chapter 16 and 17 due by Sunday. Let's see what we have due this week on the lab portion. This week we have quiz number 8 due. I hope everyone was able to complete their lab report number 2. Otherwise, please free to reach out to me after the, please reach out to me in person. Please don't flood my email. Okay, so that being said, let's get started with chapter 6. There it is. Bone physiology. Some of these will be a review of what we've been looking at in the lab. In lab, we went over classification of bones. We went over the different demarcations within bones, as well as all of these different terms. We haven't gone over the physiology of bones, like how our bones form, and that is something that we will go over today. So we're going to describe the function of the skeletal system. A lot of it is intuitive. Some of it might be new to some of you. For example, the hematopoietic function of bones. That's something that's not common knowledge. That's where blood, a lot of the blood that's circulating through you is going to be formed. And that is, we're going to look at the classification of bones. We're going to identify specialized cell types, difference between spongy and compact in terms of the microscopic structure, the different types of ossification. There's intramembranous and endochondral ossification. We're going to see what the difference between those two are. The one thing that we have in common is this transition from cartilage towards bone. We're going to look at and the effects of exercising this process of this dynamic process of bone deposition and bone resorption. That's something that from the get-go, I want us to consider and realize that bone it looks fairly stable and static, but it's constantly being remodeled by our dietary by dietary factors and also physical factors, exercise and so forth. Yeah. Every time we change our exercise routine, you will notice that effect physically within muscles. However, those effects are also happening. They're also reshaping our bones. All of these structures that we're going to talk about, all of these demarcations within bones, those are constantly being reshaped. Okay. So here we are on the skeletal system. These are the main components of it. We're going to have bone, we're going to have cartilage, as well as ligaments that are going to be part of this system. So bone, that is kind of intuitive. That's the main source of support for the skeletal system. Cartilages themselves serve as this they buffer a lot of the stress that is occurring between bones, that is occurring between bones, especially at the level of joints. And you have ligaments that are this connective tissue, this dense connective tissue between bones that is there to stabilize bones. Is there to stabilize bones in relationship to each other. And in this case, in this particular example, we observe this particular ligament, which once again, we talked about the definition of ligament is it's connective tissue that inserts and attaches, that initiates and attaches to bone, as opposed to the tendon that inserts in bone, and it's part of muscle. It joins the muscle to bone. Here we have an example of a synovial joint. In this case, there are several types of joints, and some of them are going to be more mobile than others. Synovial joints themselves, most of your mobile, fairly flexible joints are going to be synovial joints. They're going to contain a synovial cavity, and within that cavity, there's going to be synovial fluid. That makes sense, because these are your joints where there's going to be most stress due to the dynamic range of movement between them. And this fluid, the synovial fluid, is there to buffer, to really decrease the stress in this movement. See, the synovial fluid is analogous to the grease that you find in the joints of cars. And here is this cartilage also there to reduce the stress between bones. So here are the functions of the skeletal system. You're going to see in terms of support instead of storage. You also have within storage, you have calcium. Calcium homeostasis is going to be regulated in large part through resorption and deposition of calcium within the skeletal system. You also have storage of lipids, yellow marrow within the epiphysis of long bones takes its name due to the amount of lipids that are stored in that particular area. We also have blood cell production, white blood cells and red blood cells producing the red pulp, red marrow. Protection, support protection, and leverage. All of those part of this dynamic function that we mostly attribute to the skeletal system. Based on their different shapes, we're going to have different types of bones. We're going to have sutural bones, mainly found within your cranium. Here you have, there are these small bones present within the sutures of the cranium. They are, in a way, they're not consistent. They're somewhat of the remnant of endochondral ossification that occurred during development. We also have irregular bones, and we also have short bones, which are mostly cuboidal or rectangular in shape. We also have flat bones, long bones, which is your stereotypical bone, and we have sesamoid bones, which are more roundish than that are roundish in shape, flat and round. We talked about the different types of markings within bones. Here we have, in terms of elaborations, we have processes or ramuses. Ramuses, processes, there are projections or bumps. Those, I think, you will find within several bone diaphyses processes where or within regions where you have a large muscle being inserted. Once again, that usually happens at the diaphyses, meaning at the ends of bones, because that's what makes more sense in terms of physics, in terms of leverage. It's analogous to placing a door handle at the corner of a door as instead of placing the door handle in the middle of a door. Just the leverage isn't the same. And that's just due to the nature of physics. Same goes for the bones. It's like you don't insert your muscle, the tendon, you don't insert it in the middle of the bone. You insert it at the end of the bone. So a lot of my arm muscles are being inserted into this portion of the radius and ulna instead of the middle of the radius and ulna, because they can get better leverage if they are inserted in this area. So whenever I'm contracting in this, whenever I'm contracting in this direction, it's easier to pull. If you imagine the muscle present here, just pulling this particular bone, contracting and pulling this bone, it gets more leverage if the insertion is here, as opposed that if the muscle, the tendon went all the way up here and started pulling from here. If it did that, a lot of the force would be lost in the y direction here. But if it is inserted immediately where at the end of the radius and ulna in this level, then every pull will be more efficient. Every contraction will be more efficient. And so hopefully there's also, what do we have here? Openings, sinuses, those I think are intuitive. Foramens, just holes for the passage of different components. They can be as small as to allow the passage of a particular nerve or a particular artery or vein. And they can be large enough to allow the passage of your spinal cord, as in the case of the foramen magnum at the base of the skull. Fissures, we have these fissures or slits, meatuses, these canals, such as the auditory meatus, and a canal itself, duct, analogous to a meatus. Okay, depressions, you're gonna have sulcus or a fossa. Fossas are more intuitive. They look like balls within bone. Sulcus, they're grooves usually present due to the due to erosion by a particular force. Sometimes it could be a tendon or something along those lines. Okay, the rest are going to be trochanters. Those are going to be larger processes. In this case, you see it on the trochanters are associated with the femur, crest, spines, lines. Tubercles, they're more roundish than a process. Okay, the ones that will come repeatedly are going to be processes. Trochanters, you will see them mentioned within the long your humerus and femur. Condyles, you will see them mentioned often as well. They're these smooth surfaces that allow the interaction between two different bones. And trochlea is similar to a condyle, but it has more of a pulley type of disposition. Okay, let's look at based on bone structure. We have a compact bone and spongy bone. It's pretty self-explanatory, but we're going to see why they take that name. Here you have the components of a bone. You have the diaphysis, metaphysis, and epiphysis. Here you have you have the epiphysis towards the end. I've been calling that the diaphysis, I'm sure. Scratch that. So the diaphysis is going to be this middle portion, and you're going to have two epiphyses towards at the distal ends of each bone. Between them, we will have a metaphysis, which is the area where the bone will continue to grow, where this endochondral ossification will occur. That happens at the metaphysis. At some point, that area will become completely calcified, and we will start growing. But that during puberty, those cells will continue to divide and will continue to calcify, and they will continue to grow. As this area becomes calcified, it usually leaves a projection within the bone that allows us to determine whether any growth is still occurring or if that area is completely calcified. So here we have the diaphysis is going to be composed of compact bone, while the epiphysis is going to contain a spongy bone. A spongy bone, they ossify in different patterns. Spongy bone will ossify in compact bone will ossify forming these osteons, while spongy bone will ossify forming these networks called trabeculae. And this is each individual one of these lines is a trabeculae. As you can tell, compact bone is great for collecting and transferring pressures that are occurring in this particular axis. So if you think about what most of this compact bone is going to do, in this case, this femur, what most of the pressure is going to feel daily is going to be the pressure of the pressure of gravity in the y-axis, and that is what this compact bone is going to diffuse and it's going to buffer. Now, spongy bone on the epiphysis, the pressure that is going to feel is going to be the pressure of the joint, and it's going to come at all levels. So in this case, having this trabecular, this network structure will allow the spongy bone to better disseminate these pressures. Here we have within the diaphysis of this compact bone, we have this medullary cavity that will contain components for red blood, for hematopoiesis, as well as vascular components that allow the nutrients to go into the functional cells of your bone. You have your osteocytes, you have osteoblasts and osteoclasts, all cells that require oxygen, that require glucose, that require nutrients. Okay, let's look at this bone in the context of histology. The type of tissue that bone is connective tissue. What was the definition of connective tissue? It's a matrix. It's a matrix with a specialized cell types residing within that matrix. In the case of bone, the matrix is going to be these calcium salts present. And the specialized cells, these calcium salts called crystal of hydroxyapatite. Specialized cells are going to be osteocytes residing within lacunae. These lacunae will have vascularization because, once again, these osteocytes, even though they are within this calcified matrix that looks lifeless or not, it looks like it doesn't contain the structure towards maintaining cells. That osteocytes reside within it, and they will need input from the circulatory system to survive. Canalicula within this particular canalicula that you see here, these groups are going to allow passage of nutrients between these different cells. You have bone is going to be covered by a periosteum, and here we have a view of the periosteum. It contains an outer fibrous layer and an inner cellular layer. This periosteum will be present throughout all of the bone except at the level of the joint. So here we have, and here we have the blood vessels providing vascular supply to these cells, these osteocytes that reside within these lacunae. Okay. The bone matrix is going to be composed of this crystal of hydroxyapatite, and it's a mixture of both the osteoids and these crystals that provide bone with structure, but at the same time flexibility. If you look at a chalk that's going to be composed of somewhat similar components, calcium and phosphate, you will see how brittle chalks are. If our bones were as brittle as they were, we will constantly be fracturing them. So we found a happy medium within which we're able to mix collagen, this fairly flexible structural material, with this crystal of hydroxyapatite. And this component, this structure allows bone to have this calcium phosphate, inorganic calcium phosphate crystals, and this organic collagen flexible structure. So we get both. We get a flexible component and a fairly sturdy component. This is analogous to fiberglass, which is the same principle. You have you have this a very hard inorganic material, and you have a somewhat flexible matrix or flexible ground substance upon which this hardened material is present. So the osteoid, which you will hear the osteoid being referred to, is this organic component which contains proteins and collagen. Osteon is going to be deposited by osteoblasts, and this osteoid is which contains collagen, is going to calcium phosphate crystals are going to be deposited within it, and that is how you get collagen and these calcium crystals mixed together to provide strength. Okay. So here we have what I want you to get out of this is how we go from calcium phosphate to this crystal of hydroxyapatite. So you get calcium phosphate, a fairly inorganic material. One thing that one of the homeostatic systems that we have within our bodies is a homeostatic system for calcium and a homeostatic system for phosphate. So both of these are going to reside within this calcium phosphate within bones. So whenever we have a lot of calcium within our diets and an equivalent amount of phosphate, we're going to start depositing calcium phosphate within bones. However, if the calcium levels decrease, then we can start resorbing the calcium from the bone matrix. Most of the bone weight is going to be this inorganic component called calcium phosphate. So here you have your calcium, here's your phosphate. Now, what happens whenever you add, you take calcium phosphate and you add calcium hydroxide into it? So here's your calcium, here's your phosphate, and here's your hydroxide. You're going to get this hydroxyapatite, this crystal of hydroxyapatite. And the remaining, so that's going to be 2/3 of your weight, and the remaining is going to be collagen. So once again, this osteoid, this organic matrix, this organic component containing collagen, it's going to be deposited within bone, and slowly it's going to start acquiring these crystals, this crystal of hydroxyapatite, which is calcium phosphate and calcium hydroxide. And these crystals is what gives the bone its strength, and the collagen, along with the crystal, the collagen is what gives the bone its flexibility. You don't think about it as being fairly flexible, but whenever we compare it to chalk, which is also and contains inorganic calcium, that then you can see the difference in flexibility between shock and bone. So here we have the different cell types that are the principal cell types within bone. So we mentioned that as a connective tissue, we're going to have an inorganic matrix composed of this osteo, composed of collagen, and these crystals of hydroxyapatite. Now, the specialized cell matrix of this connective tissue is going to be the osteocyte, the osteoblast, the osteoclast, and a stem cell in the form of a osteoprogenitor. Let's start with the osteoprogenitor. So the osteoprogenitor is going to give rise to osteoblasts. You will find the osteoprogenitor present in this interior area facing the medullary cavity here. And you're going to have it also in the outer layer, in the periosteum. We mentioned that the periosteum contains an outer fibrous layer and an inner cellular layer. That inner cellular layer will contain osteoprogenitors as well. And this endosteum here, this inner layer first in the medulla, that contains these osteoprogenitors as well. It also contains osteoclast, this endosteum, but let's focus on the stem cells, the osteoprogenitors now. These stem cells are going to give rise, are going to differentiate into osteoblasts, and these osteoblasts are going to generate osteoid, which is going to be the organic matrix. We mentioned the organic matrix is the matrix that contains collagen, right? Collagen is organic. Calcium phosphate is inorganic. Rule of thumb, what's organic? If it contains carbon, that's what we, just as a quick rule of thumb, that's organic. Calcium phosphate does not contain carbon, right? So here you have the osteoblasts. They're going to produce this osteoid rich in collagen, and this osteoid rich in collagen can become calcified. These crystals of hydroxyapatite will bind to it and will create these calcified structures. So as this process occurs, osteoblasts will, as this ossification process occurs, osteoblasts will mature into osteocytes, and these osteocytes will become trapped within this calcified matrix, this matrix in which the osteoid, the collagen of the osteoid had basically merged with these crystals of hydroxyapatite to create this calcified matrix. All right. So here you have osteocytes residing with this calcified matrix. Now, within the endosteum, within this inner layer, alongside these osteoprogenitor cells, we have osteoclasts. These different dots here are depictions of multiple nuclei. This multinucleated cell, it's rare multinucleated cells. This is one of them. These multinucleated cells present in the endosteum, this inner layer facing the medulla, it's going to secrete acids and enzymes that dissolve the bone matrix. So this is part of that dynamic process of bone resorption and constant bone deposition. Bone resorption is going to occur whenever there is a need, there is our blood calcium levels are low, and we need calcium. Calcium is an important component within your muscles for the propagation of contractile signals. We also know that calcium is important for the release of neurotransmitters from the synaptic boutons upon the arrival of an action potential. Those are two key roles of calcium within the cell. And those roles are contingent upon calcium gradients, upon concentrations of calcium being higher in the exterior of the cell compared to the interior of the cell. If these gradients are not in place, then whenever an action potential arrives and calcium channels open, and if there's no calcium present, the gradient is not present, then calcium is not going to rush into the cell. If calcium doesn't rush into the cell, calcium is positive, +2. If this +2 signal doesn't enter the cell, then there is no change in membrane potential within the cell. Therefore, there's no change whenever an action potential arrives at a synaptic bouton at the end of a dendrite. Or the end of an axon, I'm sorry. Also, we will look in next chapter the importance of calcium within the propagation of contractile signals. Okay. So here we have osteoprogenitor cells. We differentiate into osteoblasts. Osteoprogenitor cells can be mesenchymal cells, squamous stem cells, and mesenchymal cells we will observe it. That is, we will observe it. We will mainly observe during fetal development that we have mesenchymal cells present, and we will have also squamous stem cells can be part of these osteoprogenitor cells. They're going to be important within repair during fractures, and once again, they're found in the endosteum and the cellular layer of the periosteum. Osteocytes, mature bone cells, occur in lacunae. They're connected by canaliculi, and canaliculi are the tunnels that the osteocytes travel to, but once they contact each other, they communicate through gap junctions between different osteocytes. Their functions are going to be basically maintaining the matrix that surrounds them, and sometimes during bone repair, they will undergo dedifferentiation and help with repair. Dedifferentiation is somewhat of a controversial concept. So basically, there's this notion that you have stem cells that can divide, and then you have differentiated cells. There's gradients between those two, but that is, the cells can be classified in those two terms: cells that can divide, stem cells, and cells that, if they're not stem cells, they differentiate into a particular function. So neurons, their role is to send signals hepatocytes, osteocytes, they have a particular roles, but they're differentiated cells. That's it. They have no business with undergoing cell division or returning to a previous state within their development. However, during injury, the cells take different dynamics. Cells are the way that they are due to chemical signals. The chemical signals that cells receive are going to be determined by their environment. During injury, their environment changes drastically. So that is why during injury, even though we're talking about de-differentiation, we're talking about very different circumstances, very specialized circumstances. There should be another term for this. Injury-induced de-differentiation, let's mention it that way, so that we don't think that cells can have the capacity of reverting into previous states of their life development. So here we are looking at osteoblasts. They will produce matrix. They will release osteoid, which contains which contains that was a good one. Which contains organic components as well as these particular organic components as well as collagen. And within the collagen, that is where these crystals are going to be deposited, and the deposition of these crystals of hydroxyapatite, this calcium phosphate, calcium hydroxide crystal, that is the reason why that is the process of calcification, of ossification. That is the process that gives this bone, that gives this material its strength. So right here, what it's all that this is saying is that if there's a lot of calcium, it will get deposited deposited into the bone. It's all about concentrations. There's not high levels of calcium, then there won't be any deposition of calcium occurring, because if we don't have high levels of calcium, then if we don't have high levels of calcium and undergo deposition, then we're messing with blood calcium homeostatic levels. Deposition will occur whenever there's excess calcium, whenever we have calcium within these homeostatic levels of calcium that should be within our blood for normal function of other systems, then we would not deposit calcium. Once these osteoblasts become enveloping matrix, once there's this calcification process occurring around them, then they will differentiate into osteocytes. So we have this life cycle of the osteoprogenitor cell differentiating into an osteoblast. Then the osteoblasts depositing matrix that becomes calcified. And whenever it does, whenever the osteocyte becomes surrounded by a calcified matrix, then the osteocyte will the osteoblast, once the osteoblast that releases the matrix around it becomes calcified, then the osteoblast will differentiate into an osteocyte. And this osteocyte is basically that functional cell that is whose role is just to make sure that to monitor levels and to maintain the matrix around it. We also have osteoclasts, and these are going to be present on the surfaces of bone. In this case, the endosteum of bone facing the medullary cavity. These osteoclasts, these motor-regulated cells, will resorb. They secrete enzymes and acids that allow the resorption of this calcifying matrix. And this calcifying matrix contains calcium, it contains phosphate, so these minerals will be available within, they will enter the circulation, therefore increasing levels of calcium, levels of phosphate within our blood. They are derived from monocytes. They are derived from white blood cells, basically. And they secrete these digestive enzymes and important for this homeostasis calcium and phosphate. And this is one of those enzymes, nitrate-resistant acid phosphatase. Let's look at the difference between compact bone and spongy bone. Here you have compact bone. Compact bone is going to be, it looks fairly compact except for the lacunae where you see these little holes. That's where osteocytes live. They became calcified, their surroundings became calcified, and now they exist into this, in this claustrophobic little lacunae. Each osteum is going to contain a central canal. That central canal, that is where the blood vessels and nutrients are going to come from. These osteocytes, they need oxygen, they need nutrients, and these blood vessels are going to supply it. So that is going to be compact bone. Spongy bone, on the other hand, the calcification process, here you observe a compact bone in which basically calcification happens in a dense manner. On the other hand, the spongy bone calcification happens in this tube-like like fashion, where this level of calcification gives this bone a level of hardness, of structure, but at the same time a level of flexibility. Each one of these tube-like components is called a trabecula. And in this cross-section, we can observe that you see this ossification pattern occurring similar to that of a compact bone, in which these osteoblasts are producing matrix, and eventually that matrix become calcified, and the osteocytes become trapped between these lacunae. As you can see, this exterior is going to be the endosteum, and this endosteum contains the progenitor cells as well as osteoclasts. What do we have here? So there's going to be there's going to be 3 types of lamella. And their names are going to be just determined on their structure, their orientation. Surrounding the bone, you're going to have circumferential lamella. Surrounding, you can think of it surrounding the circumference of the bone. That's going to be your circumferential lamella. Between each osteum, there's still some lamella. It doesn't have that much structure, but it is lamella nonetheless. That's an interstitial lamella, inter in between, and surrounding each osteum, and that's what is known as this concentric lamella. At the center of each osteum, we're going to have a central canal. That central canal is going to provide these blood vessels. Between each osteum, there's going to be these perforating canals. So in order for these blood vessels to reach deeper into the osteons within the most inner portions of this bone, they're going to traverse through these perforating canals. Okay. So here is, I think we touch slightly on the benefit of having compact bone within the aphysis of this long bone. This compact bone allows the diffusion of pressure coming from this direction, the direction of gravity, and the direction of gravity that's pulling our body weight, and that body weight is being transferred through these long bones, in this case, the femur. So here we have most of the pressure is coming in this direction. Rarely does the pressure come from any other direction. I guess if you get hit, the pressure can come from another direction. And this long bone is mainly adapted to withstand pressure, unidirectional pressure, and an impact can shatter this bone due to this. The disposition of the osteons, the orientation of the osteons. On the other hand, you have the spongy bone, which is better suited to handle pressures coming from several directions. And that would be akin to the pressures that are occurring in this particular joint. All right. Let's talk about the periosteum. The periosteum, its role basically is once again, it contains two different layers, a fibrous outer layer and a cellular inner layer. We talked about how tendons and how ligaments, how much pressure they undergo, and that pressure is bones serve as an anchoring point for these components. So every time you flex, every time we lift something, those muscles are pulling on bone, and we can do tremendous feats of strength, and that muscle it's able to perform that strength due to the how strong the bond or the linkage between this bone and tendon is. This is due to the collagen fibers in the outer layer of the periosteum being contiguous with the collagen fibers of tendons or of ligaments. They're one and the same. So at this level, they're basically fused together, the tendons of muscles and the tendons, sorry, the collagens of the collagen of tendons and the collagen of the periosteum of bones, they're fused together, they're the same collagen. In that sense, this periosteum is contiguous with tendons and joints. There is also this perforating fibers perforating fibers that will reach farther past the periosteum and into the circumferential lamella of bone. This circumferential lamella present here. It contains the fibrous layer will contain fibroblasts. As any connective tissue, fibroblast is going to be the one that makes the fibers, right? And collagen is a fiber. Any connective tissue proper. And the cellular layer will contain these osteoprogenitors. So let's talk about the endosteum. Let's talk about this inner layer of bone facing the medullary cavity, this inner layer lining every single trabecula within a spongy bone. Here you have this this view of endosteum containing an osteoclast. Here are the osteogenic cells, and we also see osteoblasts within the endosteum, but the main functional cell types will be the osteoclast and the osteoprogenitor. The osteoclast is going to be just resorbing calcium phosphate, resorbing this mineral. Is there for growth, repair, and remodeling. It will line okay, so I think I repeated all of this. Endosteum present in the inner layer, also present in trabecula, and there is going to be any part that's incomplete is going to be due to this osteoclast having access to the calcifying matrix for resorption. All right, let's save bone development and growth for next class, okay? We're going to finish this up on Thursday. Do we have any questions or any requests? No? All right, well, everybody enjoy dinner, enjoy the rest of your day. Bye.
Bye, thank you.